Gene expressions. Gene expressions can be represented through two main processes, transcription and translation. Let's begin on the basics. Gene is known as a functional part of DNA or just a sequence of DNA. DNA, on the other hand, carries inherited genetic information using the body's growth system for its functions. The function of DNA is to code for protein, therefore gene codes for protein. Now, to understand how gene can be expressed, we will also need to understand the structure of DNA. DNA structure is made of many nucleotides. These nucleotides are then made up of a sugar carbon, a nitrogenous base, and a phosphate group. For DNA, there are 4 different types of nitrogenous bases: guanine, cytosine, adenine, and thymine. As a side note, RNA has almost the same nitrogenous bases as DNA, except that uracil replaces thymine. Moving on, one of these nucleotides are joined by another nucleotide through a bond between one of the sugar carbon and one of the phosphate group, known as the phosphodiester bond. All of this forms a polynucleotide. The sugar and phosphate units make up the backbone of this nucleic acid and showcases one strand of DNA. As one would probably know, DNA is double-stranded. This works due to hydrogen bonds existing between the nitrogenous bases. Just a reminder, hydrogen bonds formed between hydrogen and a more electronegative atom or group. They are weak individually but collectively very strong. And now we have our double helix DNA structure ready to be transcribed into messenger RNA. Transcription. Transcription is the process where a copy of DNA is made in the nucleus, and messenger RNA or mRNA is the copy of this DNA strand. Transcription starts with the DNA being unwound with the help of an enzyme, RNA polymerase, which binds to the promoter region of a gene. Therefore, the hydrogen bonds between the bases slowly break apart, which causes the two strands of DNA to separate. Then, 3 RNA nucleotides find their complementary base pairs on the DNA strands, and hydrogen bonds slowly begin to form between the two bases of the RNA and DNA. Eventually, the enzyme of RNA polymerase then catalyzes the formation of phosphodiester bond between the ribose sugar and the phosphate groups of the adjoining RNA nucleotides. And there you have it, the mRNA molecule is formed. However, the mRNA contains some non-coded regions that must be removed. These are known as intron parts. A group of 5 snRNPs, or ribonucleoproteins, will come work together to remove only the introns. Translation. Translation is the process of how the mRNA molecule is read and thereby translated into a protein. After transcription, the newly formed mRNA will now leave the nucleus through a nuclear pore and enter the cytoplasm. There, a ribosome will become attached to the mRNA. Now, 2 new RNA molecules will come in place soon. They are the ribosomal RNA and the transfer RNA. The ribosomal RNA makes up ribosomes, while the transfer DNA matches amino acids to mRNA to help make proteins. Remember, the transfer DNA is the most crucial part of translation. Going back a bit, the genetic code of the mRNA is read in groups of 3 letters called codons. Our codon is often referred to as the 3 nucleotide unit on the mRNA strand. Our codon codes for amino acid by translating data through an anticodon to the transfer RNA. The following is a table of all 3 letter codons with the related amino acids. Multiple codons code for one amino acid to avoid DNA mutation consequences. The transfer RNA now attracts itself to one of the codons. The tRNA contains a complementary anticodon in the front, planning to connect to the first codon of the mRNA. There's a hydrogen bond now between the nitrogenous bases. Eventually, more and more tRNAs will come being attracted to the codons with their complementary base pairs spinning alongside the specific amino acids that they code for. As this process continues, peptide bonds are formed between amino acids creating polypeptide chains. For now, just remember that polypeptide chains is just really a protein and it has peptide bonds within itself. While this process is ongoing, once a peptide bond has been formed, the former tRNA who translated the codon to the amino acid is now allowed to leave to find another codon of a different mRNA to translate. And that's all. A protein has been successfully formed through 2 major steps of gene expression. In the end, the protein will be exported out of the cell through a vesicle from the Golgi apparatus. The importance of protein in your body and cells. Starting off with the body, protein is required for the maintenance and growth of tissues in the body. Protein is broken down in the human body in a quantity equal to that which is consumed for tissue growth and repair. The body occasionally breaks down more protein than it produces, increasing one's requirement for it. Moving on, enzymes are a form of protein that speeds up metabolism in the body. Metabolism is a general term for all the biochemical reactions that occur in a cell. Body functions that depend on enzymes are digestion, energy production, blood clotting, and muscle contraction. Some proteins are hormones, which are chemical messages that help your cells, tissues, or organs communicate with one another. They are produced and secreted by endocrine glands or tissues, and then your blood carries them to other tissues or organs that they are intended to affect. There they bind to protein receptors on the cell surface. Common examples of hormones that you may know are insulin and glucagon. Proteins also act as a buffer through one type known as the hemoglobin, a protein that makes the red blood cells. A buffer is a substance that can stabilize the pH of a reaction. Hemoglobin utilizes small amounts of acid to retain this equilibrium, allowing us to have stable body temperatures. Cells are supported and given structure by proteins. Additionally, they permit the body to move on a bigger scale. This is done by a class of proteins known as the fibrous proteins. There are even transport proteins in our body carrying nutrients in and out of our cells. These nutrients include vitamin and minerals, blood sugar, cholesterol, and oxygen. Linking up with the previous topic, transport proteins carry vitamin into the cell to give to the enzyme to help catalyze the biological reactions in the cell. Overall, the body uses protein for a variety of beneficial purposes. The impact of one's environment and lifestyle on gene expression. To recap, gene expression is a process through which the gene's information is used to produce either non-coded RNA molecules that perform additional functions or RNA molecules that code for proteins. Since this comes from the DNA, the code is hereditary, yet external forces can indeed influence one's expression of gene. One's experience in their lifestyle can easily influence gene expression. The chemistry that encodes genes in the brain cells can change either temporarily or permanently, depending on whether positive influences such as exposure to visual opportunities or negative influences such as malnutrition or environmental contaminants have had an impact. This process is known as epigenetic modification. Epigenetic modification happens when a human is exposed to external forces such as drugs, harmful food, and pollution. These detrimental substances enter our body, threatening the process of transcription and translation, forcing our body to respond by turning off certain genes, weakening us. On the other hand, epigenetic modification can be positive as warm temperature, good sunlight environment will bolster better human structure foundation through turning certain genes on. A comfortable lifestyle with a good nature environment and good diet can easily strengthen our body through the influence of the gene expression. This is really due to a strong supplement of vitamins and nutrients going into the body. A quick example of positive epigenetic modification is sunlight, as that gives a person vitamin D, strengthening their bones. A quick example of a negative epigenetic modification is pollution, as dirty air increases the chances of respiratory infections, heart disease, and lung cancer. The history of genomics. The history of genomics dates back to 1871, where Friedrich Mischler discovered the presence of DNA. In 1904, Walter Sutton and Theodore Boveri discovered that chromosomes are found in matched pairs with one chromosome coming from each parent. In 1910, Albert Kolsel won the Nobel Prize in Physiology for identifying the 5 nucleotide bases. From 1950 to 1968, many scientists rapidly developed the knowledge of DNA and RNA stranded in our nucleoplasm. Frederick Sanger created the SARS X174 virus in 1977 and used it to sequence the first complete genome, opening up new possibilities in genomics. From 1983 to 1998, scientists quickly invented new techniques into analyzing genes, being able to read them and how their functions operated in the cell. Going back a bit to sequence the entire 3 billion letters of a human genome, the Human Genome Project was established in 1990. In 1999, the project's initial chromosome, chromosome 22, was sequenced. Humans have 20,000 to 25,000 genes according to research, which was finished in 2003. In the present, the field of genomics known as person genomics focuses on the sequencing analysis of a single person's genetic information. It is as if the Human Genome Project was conducted specifically on one person. Off of the person genomics, there has also been research into genetic engineering as a result of the consequences of the Human Genome Project, where humans can somewhat change their real-life stats. Recombinant DNA. Recombinant DNA, a form of genetic engineering, takes DNA from 2 distinct species to put into a host organism to create new genetic combinations used for science, medicine, agriculture, and industry. With the use of recombinant DNA technology, it is possible to isolate a single gene or DNA segment to allow researchers to learn its nucleotide sequence. Because of that, we can get the DNA of interest we took out from a cell to place it in a plasmid, a small circular strand of DNA, to form a recombinant DNA strand with the help of an enzyme, DNA ligase. The recombinant DNA is then inputted in the bacterial cell, where it will then copy and divide itself. Recombinant DNA has many uses. For instance, recombinant DNA can help crops become more resistant to disease and yield more. On the other hand, recombinant DNA can boost the production of antibiotics and vaccine, important in medicine and lowering cost. My personal favorite is how recombinant DNA can enhance nutrient amounts in the food while guaranteeing the safety of the product.